Before we're ready to start, I want to quickly introduce the format of today. This is the 6th edition of the uniMIND Central Sessions, which is a monthly meeting with the authors of selected papers in the topic of psychedelic research and sometimes consciousness research. And it's an interactive format, so there is a structure to it, but everyone is free to join in and ask any questions that pop into your minds. And we will also, of course, try to answer them to the best of our knowledge. One other thing about this format is that we talk about the content of the paper, of course, but it's also about what happens around the paper. So about the people who wrote it and also the processes behind it. So we will also briefly touch into that. All right, so we are now around 10 people in this Zoom meeting. I will slowly start the session. Right. So at first I asked if people have a science background. Around 3 people raise their hands. Now I'm curious how many of the people here have a background in the fields of neuroscience, cognitive science, or biology, psychiatry. You can show your hands. Okay, 4 people so far. Good. So now, yeah, we have an idea of, I guess, the depth in which we will go into with some concepts, but also keep in mind that not everyone here has a science background, so we'll try to explain the things that are a bit more hard to grasp or require a bit more background. All right, so I would like to briefly introduce myself. My name is Vlad Nicolescu. I have a background in biochemistry and cell biology with a master's in neuroscience. And I worked on projects on the endocannabinoid system. And later on, I did some research on human metacognition and imagination. And of course, my big interest right now is psychedelics. And I've been working at the Mind Foundation for the past almost 2 years. And today with us, we have Kacper Łukasiewicz. I hope that's how you pronounce your name. Who is a co-author of the paper that we will talk about today. So Kacper obtained a master's degree in biotechnology from the University of Warsaw. Then he worked with mouse models of alcohol use disorder at the Nęcki Institute of Experimental Biology of the Polish Academy of Sciences, where he obtained a PhD in neurobiology. Recently, he completed a postdoctoral fellowship at the University of California, Santa Cruz, where he studied mouse models of autism and psychedelic-induced behaviors in mice. He specializes in behavioral research and analysis with machine learning tools. And now he is leading a research laboratory in the Psychiatry Center at the Medical University of Białystok. So welcome, Kacper.
Hello.
Today we will be discussing one of your papers, namely Serotonergic Psychedelics in Neural Plasticity, which appeared in 2021 in Frontiers in Molecular Neuroscience. So before we get into the paper itself, as I said earlier, I would also like to get some insights into the things that were around the process of writing this paper. So what was your personal motivation for co-authoring this review? What draws you to the topic of neuroplasticity and psychedelics?
Mm-hmm. Yeah. So firstly, like the neuroplasticity topic field is quite close to my previous research done on my PhD. So I was always looking like from this perspective. So what's happening with the neurons, especially at the synapses, at the dendritic spines, and Zoo Laboratory, where I was doing my postdoctoral fellowship, is specializes in imaging of synapses. So we had a some projects going on about imaging of synapses. And one of the ongoing projects in the lab was on psychedelics. So we were using classical psychedelic, the DOI, so dimethyloxyiodoamphetamine, as a classical psychedelic, and we were testing some of the newer analogs of the ibogaine, namely TBG, so terbinafine. And because I wanted to get involved more in this project, I thought that a new good way to go into it would be to review the literature, to search like how actually these compounds may work on the plasticity, and I found that this knowledge is pretty scarce and incomplete. So I thought that it's a good idea to gather it and write some short review about it. So we just started writing a paper about what's known in the field, and we realized that there is not so much known clearly about how these substances work.
Right. So you also have more intimate knowledge of the substances that you mentioned in this review. I'm also surprised that you mentioned TBG. It's something that I have a personal interest in. For the people who are not familiar with this, it's an ibogaine derivative that is supposed to enhance neuroplasticity in mice without eliciting the psychedelic-like behaviors. So this is something that I would like to come back to later on. Also, for tonight's participants, especially for the people who don't have a specific background in these fields, I was wondering, could you provide a brief explanation of what is neuroplasticity and what kinds of neuroplasticity are out there?
Yeah, so how to say it briefly? So neuroplasticity is a term describing plastic changes of the neurons, which are base for the network formation. So we have many neurons, they are connected together, the informations flow from one to the other, and they form like the networks of and circuits. And the connections between them are modulated, the strength of them, the formation and elimination of these connections, and the process of these plastic changes of the connection between neurons is what we call the neuronal plasticity. And we can think about it at multiple levels of complexity. So one way to look at it is called cellular plasticity. So we can have changes of a particular like neuronal properties. So the neuron itself may produce some proteins or degrade some proteins. So it can change its content, which affects both the size of the neuron and how it works. So we can say that it's a kind of a cellular plasticity, but also this kind of the most general plasticity involves the formation of a new neurons and the modification of a newly formed, like the way of specific forming a new neurons and making them work in a particular way in a network. And when we go down into smaller pieces of the neurons, we can say that that these changes of a particular compartment of the neuron may be called a structural plasticity. So we have a change of a structure of a neuron, and we can distinguish structural plasticity, which involves the plasticity on axons of a neurons, dendrites, so like a dendritic structural plasticity, and very often we talk about the structural plasticity of the synapses because these are very labile structures. They change their shape very rapidly, and also they disappear and they are formed to form a particular connection with a neuron. So the changes of synapses are called like synaptic structural plasticity, and the synapses themselves can differ in shape and size, but they can also function in a different way if they have changes in their electrical properties, which are based on the chemical properties of the proteins, which are based on these synapses. So we can have a more receptors or more receptors which are less or more conductive. And even without the changes in the structure, we can have changes in the function. So then we call it a synaptic functional plasticity. So just repeating this, we can think about the plasticity at the level of the cell, at the level of the compartments of the cells, especially the synapses. And when it comes to their shape, we talk about structural plasticity, and when it comes to their function, we would call it a functional plasticity.
I see. So plasticity is something that's basically pervasive at all the levels of brain functioning. And what would you say is the physiological function of these processes?
Formation and elimination of the connections between the cells. So as we know, like the brain is a plastic system, so it needs adaptive changes. So we need to form new connections. We need to maintain connections which are important, and we need to eliminate those which are not important or which are need to be eliminated to rebuild the memories or to form new ones, or to erase the ones which are not needed anymore. Yeah,
I see. Yeah, it's interesting how psychedelics as a class of compounds seem to have this this very broad effect on these kinds of neuroplasticity. I was wondering, from your own experience doing your own projects on this topic, how would you go about studying neuroplasticity in mice or neural cultures?
So there are a lot of approaches. So the my favorite one is called in vivo imaging of structural plasticity, and also in vivo imaging of functional plasticity. So the one where you directly look at the brain at the level of the neurons, so like like cell bodies, or at the level of the synapses. And when animal have some experience, you observe how the experience affects the synapses in a particular brain region. Because I think like this approach gives you the most information how actually your task is related to brain structure, but it's also a very demanding approach, and it needs quite a lot of expertise, and some of these approaches between each other are exclude themselves, are excluding themselves. So like, for example, in a lot of these tasks, when you want to observe a synapse under the microscope of a living animal, you have to put it under the microscope, so it has to be head fixed. So you are restricting the natural behaviors of the animal which is normally moving around. So you're putting this artificial environment, kind of behavioral model, because it's not a naturalistic behavior, but you are modeling a behavior, and you are looking how close your model is to the processes which we believe are like universal also outside of this model.
I understand. So if you would study psychedelics, you would use the same probably paradigms, but just also administering psychedelics to these mice in different conditions. Someone from the participants asked, would it be fair to say that noradrenaline capacity to recalibrate addiction or reward-oriented neurobiology is a result of its neuroplasticity potential, or would it be the other way around?
Could you repeat? So we are specifically going into yeah. So could you repeat the question, please?
Yeah. So we're talking about noradrenaline and its its effect on addiction, and it says that the question asks whether noradrenaline's capacity to recalibrate addiction is a result of its neuroplasticity potential, or would it be the other way around?
Actually, we don't know why the ibogaines or like noribogaine affects addiction. So we don't know exact mechanism, but for sure it affects the like the brain plasticity. But the problem with the addiction when it comes to the like explanation of why it works on addiction is that there are a lot of brain structure involved in the formation of an addiction, and we can collect some evidence about the particular brain structures affected by the particular psychedelic, for example, ibogaine or noribogaine, but it's not easy to say why actually it affects the addiction in general. So it's a composition of many actions on many brain structures, and sometimes they are like, I don't know, happening in the other way. So like in one brain region we can have a plasticity, like formation of a synapses. In some other we can have an elimination, and it also depends on the type of the neurons. So like the whole brain also is composed of excitatory and inhibitory cells, and we may think about it as the like increase of the excitatory neurons firing as something positive, which will lead us to the like, I don't know, enhancement of the memory. But we may think, okay, we need to increase the activity of the excitatory neurons and decrease the activity of the inhibitory neurons. So then we will boost the brain activity, or we will get some effect, for example, like anti-addictive effect. But it's not that simple, because like generally, if we just stimulate the brain, we would have an like increase in the activity, and probably we will end up with seizures. So many of these processes involves also the balance of excitation and inhibition, and a lot of these processes like very subtly modulate how neurons in a particular brain regions work together. Yeah.
All right. So thank you for that answer. Before moving on to the bulk of the paper, I would like to go a bit back again into the methods regarding the question I asked you before. Raven would like to know in which brain regions of the mouse would you use in vivo imaging of structural and functional plasticity? Would you mind giving a few more details about the methodologies?
So it's not about this paper in particular when it comes to in vivo imaging, but it's an interesting question, and I would love to answer it. So in general, you want to target the brain regions which are on the surface, because you have limitations when it comes to the depth of the imaging when you use the optical imaging. Because like if you want to go deeper into the brain, then you would use the electrophysiology. So then you can put your like probes, your electrodes in any brain region. But for the in vivo to photon imaging, you need a microscope put on the like objective has to be close to the skull, and you have a very limited depth of the imaging of the brain. Depends on the method of the imaging that you use, but in general you would have to look for the cortices, brain cortex, and the region of the cortex which are accessible on the top of the skull of the animal. So in theory, there you can also access some regions which are deeper, but it's much harder. So, for example, like the prefrontal cortex in mouse is like deep into the anterior part of the brain. So it's not easy to look at it from the top of the brain. Then you have to like look from the side of this like internal piece of the brain. So in some papers it was done, and sometimes you can go with your model and try to look at the part of the cortex which are on the surface. Like for example, in the Alex Kwon's paper on the prefrontal cortex and the activity of the psychedelics on the dendritic plasticity in the prefrontal cortex. But there are like limitations. So in general, the parts of the cortex which are on the surface of the brain. So the somatosensory cortex, motor cortex, visual cortex, parts of the prefrontal cortex, but not too much with the entorhinal cortex. Hippocampus is accessible, but with some additional surgeries. And for example, looking into thalamus or into auditory cortex is technically possible, but it's very hard.
I see. So it is rather lucky that a lot of the effects of psychedelics are allegedly from activity that they influence in the cortical surfaces in the prefrontal cortex. So now going back to the topic of psychedelics in particular, I had some questions reading your review. Most of the questions are related to the actual papers that were there, but I'm also interested in a broader view and connections with the things that are not explicitly mentioned there. So there is one thing that drew my attention in your review when you were talking about 5-MeO-DMT. You mentioned that, quote, it has been speculated that 5-MeO-DMT's action may depend on brain states. So in anesthetized animals, it preferentially targets the 5-HT1A receptors on GABAergic inhibitory interneurons, whereas in awake animals, it primarily binds to the 5-HT1A receptors on excitatory pyramidal neurons. Now, I'm not sure if in the paper there was more discussion on this, but this to me reads as there would be some sort of hints towards the idea of set and setting as kind of determinants of brain states. So the set and setting of ingesting a psychedelic compound in humans would determine the state of the brain that the person is in when the psychedelic is ingested. And as we know, these can have a great impact on the experience itself, and later on the therapeutic benefits. So talking about neuroplasticity, do you think there is some sort of interaction between set setting and neuroplasticity? Would it be differentially affected by how and where a psychedelic is ingested?
Okay, so yeah, so you mentioned an interesting piece of the paper. That was actually one of the hardest part to write because it's based on, I think, like 2 or 3 papers which are in some way contradicting each other, and it is not well like discussed why. So we were like thinking how to interpret this and what actually it might mean.
I see.
Yeah, because as I said, there is not so much written in the field of the neuroplastic effects of psychedelics. So also there is like a lacking discussion, and a lot of topics are not covered. Some of the topics are like even I don't know. like it comes from a paper, but it's actually not discussed. So I think like some people are just omitting going into some field because they know that there is not enough support to defend the paper from the reviewers. So they don't want to go into it. Yeah, it seems that there is a lot going on when it comes to the set and setting and in general the brain state. So we know that, as I said before, there is a kind of like a balance between the different types of the neurons, and the activity of a particular neuron depends on its activity, its stimulation by some other neurons, but also a lot from modulation. And this modulation comes from many neurons with a modulatory neurotransmitter. For example, a lot is done by the dopamine, and we know that the psychedelics stimulate the dopamine receptors, but we don't know what's the function of this stimulation and how it affects the plasticity in general. So that's one aspect which is of the psychedelic action, which is now studied. It seems that it has to be studied in the coming years, because like from the recent paper from the field of depression, we also know that the dopamine modulation is extremely important when it comes to the action of the antidepressive drugs. And we know that the psychedelics, one of the most potential therapeutic potential of the psychedelics is their antidepressive action. So it's probably at least partially through the dopamine receptors. So this state is as I said, is regulated by the neurons themselves, by their inputs, but also by the modulatory neurotransmitters in the whole network. Of course, there is also an aspect of the glia, which is totally not studied when it comes to the psychedelics. I think, like, I don't know if there are any papers on the effects of psychedelics on the glia, but we also know that the astrocytes can modulate the threshold, the activity of the synapses in the whole networks. So that's something which has to be studied in the future. And when you ask about the set and setting and in general and the plasticity, I think that's one of the most important discussions in the field which is going on now. So we don't know if it's enough to just stimulate the neuroplasticity to have the therapeutic effects of the psychedelics. If so, then we can use the psychedelics which are not hallucinogenic, and we can have the therapeutic effects of them. So in theory, in future, we can go to the drugstore and just buy a pill which is non-hallucinogenic and would have the same effects, neuroplastic effects, as the psychedelics. And that's an approach which is developed very strongly by the David Olson's lab from UC Davis. So very like promising type of these analog of the psychedelics is actually the TBG. So we have a compound which have a similar affinity to the receptors as ibogaine, but it lacks its cardiotoxic effects and it lacks hallucinogenic effects, but it keeps the anti-addictive properties and stress releasing properties. So that's one approach. And the other approach is quite on the other side, and it's more into the history of the use of the psychedelics as a tool in psychotherapy. So we have a psychedelic assisted psychotherapy. So by definition, the psychotherapy is more important than the psychedelic. The psychedelic is a tool, and it gives us the chance in the particular set and setting to open for the therapy to be able to actually rewire your brain and change your like, I don't know, change your thoughts and change your mind in a way that it's therapeutically useful. And in this other approach, you may not even don't care about the neuroplasticity. So if you have this kind of very strong experience which can rebuild your personality, dissolve your ego in some definitions, then you don't even have to care if they are neuroplastic or not. You have a event which is changing your life.
A revelatory experience.
Yeah, and I think that for me the most reasonable approach is in the middle. So it's a lot of researchers from the Johns Hopkins University believe that actually you have a brain plasticity going on in the brain, but in general it's more specific to the young age than to the adult age. And when you are adult, you have a closed window to some of the plastic changes, and if you are in the state which is pathological to some way, like some social pathological state or like addiction state, there is not so much room for a change, because the brain is not as plastic as it was in the future. But some processes which are going on, as such as addiction, may rely on the period when you were younger, and something happened and actually led you to this state when it is not easy to change it. But the psychedelics will open up this window for the plasticity and allow you to change this pathologic state, because you have enhanced plasticity. But if you just increase the plasticity, nothing will happen, because you need to actually give a therapeutic intervention to actually change it when it's possible, because you opened plasticity.
Right.
And I think it makes the most sense from these two approaches, but like both of them and have some pros and cons. But I think this middle one is the closest to the therapeutic use of the psychedelics that we can have in the future.
I see. So in this view, there are multiple things going on at the biological level and also at the psychological level, and you would possibly conceive that there are compounds that just increase the neuroplasticity without having any sort of corresponding experience, such as the one elicited by psychedelics, and that then which can be used for the work in psychotherapy, and then potentially have an effect that is also long lasting. But it depends on the work that's done after this window of neuroplasticity is being open. What I do find interesting is that a lot of people refer to experiences that they've had during these psychedelic experiences, to specific memories, specific maybe traumas that came back in those moments, and then working on those is the thing that helped these people improve their condition after all. So it seems that to me at least, it seems that there's also something about the acute experience that tends to at least be more specific in terms of the kinds of experiences in the past that triggered the ailment, and that needs to be discussed with the psychotherapist. So you talked about these compounds that could in principle increase neuroplasticity, and if you would give this compound to someone, there would be some increase in neuroplasticity in specific brain areas in the prefrontal cortex, for example, in the hippocampus. There is a lot of talk in the papers that you covered about how psychedelics and also non-hallucinogenic psychedelics increase plasticity. So they increase the complexity of the dendritic arbor. They increase the number of dendritic spines, number of synapses, um, in terms of the survival of the connections, because it seems that ultimately it matters how many of these connections survive in the long run and are sustained over time, rather than just how many new connections appear. What can you say about the survival of the connections induced by psychedelics? Do they last the test of time, or is it just an indiscriminate increase that will eventually fade away?
Yeah, that's a good question. Yeah, and when it comes to the like long-lasting effects, actually there is much less done in this field than when it comes to the acute effects, because
Mhm.
of like many reasons. Like it's of course it's much easier to do it in the acute models, because like also they're easier to run, and also a lot of models by definition are acute. So for example, like maybe not by definition, but in the, you may say about the acute effects, acute and long-term effects. So some people distinguish between the acute effects which are happening like in minutes to hours after the administration of the drugs, like subacute effects, which are like around days, but like up to week, and some long-term effects which are long-lasting more than a week when it comes to weeks or years. But also the definition of the acute and long-term effect depends on the field. So in the cell culture, you would define the acute and long-term effects differently than when it comes to the animals or when it comes to the human clinical studies. That's a general comment, but as I said, there are like much less research on the long-term effects than on acute. And I agree with you that you may have an effect when some of these drugs have very spectacular effects acutely, but actually nothing is going on in the long run.
Mhm.
And the question is if it comes from the because of the model. So maybe nothing is done which would support these changes to stay in the brain. Maybe some of these drugs just have an acute effect, which is not long lasting, which makes them not so useful as a therapeutics. In general, we know that the psychedelics are more long-lasting than some of the other drugs. So, for example, we know that a single doses of the psilocybin or DMT are quite effective in the long run for their therapeutic effects in humans, compared to the, for example, ketamine, which is not a classical psychedelic, but it shares some of the properties with the psychedelics. But we know that for ketamine, we need multiple administrations to be effective as a antidepressant drug. And also we know that the standard drugs such as selective serotonin reuptake inhibitors have this con of like this bad side of needed to be administered repeatedly, and also producing some tolerance in time, which needs adjustment of their doses and so on. So there is a promise when it comes to these long-lasting effects of the psychedelics. There is not so much research actually from the basic research proving that they work in the long term. One was done by our group, so by group of ISUA and we it's paper by Zhu Lu and Michel and Michel, and I was working with the in this study, and we actually found that in a model of the chronic unpredictable mild stress, which is one of the models, animal models, which have a pretty good validity when it comes to the models of depression in rodents. A lot of the effects of the TBG, so this synthetic analog of ibogaine, were long lasting. So the animals were had like a lot of behavioral features after the TBG treatment, which were long lasting, and also like the cell activity was normalized after this drug. So I can say that I believe, I know that these drugs might be long lasting and that they have a very they are like very promising, but we need much more research to actually say it's say it for most of them. Yeah,
I see. And as far as I know, no human has ever ingested TBG so far. So
I don't know that, and I haven't heard about it yet.
Right. So indeed, it does not elicit the head twitch response in mice.
Yeah.
But I'm wondering whether that truly means it's completely non-hallucinogenic in humans, because there are sometimes some discrepancies between the head twitch response and the actual psychedelic effects.
Mm-hmm.
If I'm not mistaken, 5-HTP elicits. 5-HTP is a serotonin precursor that is not psychoactive in humans and does elicit the head twitch response in mice. So in the end, that's also one thing that we need to find out, if these compounds are indeed non-hallucinogenic.
Yeah, I agree. And yeah, that's one interesting thing that your comment mentioned, like touched, is how bad are our models of the like animal models for the like for the psychedelics. So as you said, like the closest behavioral proxy that we have for the hallucinations are the head twitch responses, and it's a behavior which is quite specific for mice, not too much for rats. So rats have a different behavior.
Could you briefly explain the head twitch response to the people who may not be familiar to it?
Yeah, so from animal studies, we know that classical psychedelics, which from the anecdotal evidence, but also like from the evidence which we know that are hallucinogenic for humans, are inducing the head twitch response in mice. So in general, the mouse is like shaking the head. So normally the mouse is like moving around and moving the head in the arena. But after the psychedelic, it goes and it shakes the head like very rapidly and from time to time. And we may believe that it's a way of like having a hallucinations of the mice, for the mouse, because like if you think as a mouse and you start seeing the things which are not there, one way to check if you're hallucinating is actually like to check for surroundings and make sure that what you're seeing is there. And so like shaking the head is a way of like checking the environment, and so it is a proxy for hallucinations. As I said, for substances which are hallucinatory for humans, mostly they are provoking head twitch response in mice. Also, the substances which are stronger when it comes to the hallucinatory effects in humans, they produce more head twitch responses. But as you mentioned, not all substances works this way. So this is just a model of the hallucinations, and also it is not very close to our hallucinations, because we also know that the hallucinations differ in quality. So we can have like visual hallucinations, or auditory hallucinations, or sensory hallucinations, and we don't know which of these is modeled by this behavior. So it's a rather a correlative behavior than the behavior which we understand. And that was one of the points of my research during my postdoc. So to look closer into this behavior and try to say actually what's going on there.
That's true. Well, so far it's the best and the most efficient way of the best we've got in terms of assessing these effects in mice, and the most also cost-effective way of doing it in the laboratory. An interesting question came up in the chat. I'm trying to keep up with the chat, but it's sometimes hard to both read and speak. So people can also just raise their hand or just intervene if they have anything to say. I'll read this question because I find it very interesting. Although neuroplasticity is something we usually consider as desirable, at some extent it might be prejudicial as we need stable representations of our world for our mental health. Do you think that neuroplasticity induced by psychedelics or non-hallucinogenic psychedelics can be harmful at some point?
Mm-hmm. Yeah, I totally agree. That's what I said some time ago. So I believe that we need a particular states of the brain, and sometimes we need to be more plastic. Sometimes we need to have more stable memories, for example. So if we have a very increased neuroplasticity, it means that we are also forgetting much faster. So it is not always desirable to have this kind of plasticity. So I think that's why you have to be very wise in implementing these substances into clinic and into the regular use, because in general they may harm a lot of people, but there are groups of people who, after a clear like medical examination, could have a very high chance of having a profit in their mental state after using the psychedelics. So yeah, so I think they need to be particularly suited for patients.
That's a very important point that you're making, I think, because as you said, SSRIs and also the treatments that we have so far that work on serotonin reuptake also elicit this neuroplasticity, and these are usually taken over long periods of time, which means that this window of increased neuroplasticity is open at all times, which could, as you said, have effects on memory, and which is not always desirable. So we would it would be another reason for why someone would benefit from just a few of these experiences spaced in time. So Dasha has a question for us.
Yeah, hiya. So I was just wondering, obviously a lot of the research that's been done so far is looking at like the structural effects in terms of neuroplasticity. I was wondering how much we know about whether that is then kind of related to like the change in strength of synapses, so like more functional, like how much sort of electrophysiological research has been done in this area.
I think there is much less done in the functional field than in the structural field. Like from our research, it seems that it's, I don't know, 1/3, 1/4 is more functional than compared to the like about 3 times more structural research. Yeah, but there is also some research going on. Like as I mentioned, this paper by Zhu Lu, which is the first author, which is also from the, I think, last year. So we've also done the functional imaging, so the activity of the neurons, and we found that like some activity pattern which was disrupted by the model of the stress is normalized when you use the analog of psychedelics. So you can have like a normalization of the activity pattern of the neurons. But there are like some of the electrophysiological studies, but I agree that there is much less there than when it comes to the structural plasticity.
All right. Thank you, Kacper, for that answer. We are slowly approaching the end of the session. There were some things that were mentioned at the end of your review that I found interesting. And I was wondering, based on those questions that you posed at the end, what do you think are the next steps in preclinical psychedelic research? And not necessarily terms of neuroplasticity, even though, of course, you can also talk about that. But what do you think is the most pressing question that we can solve with preclinical research?
Mm-hmm. Yeah, I think the problem is that there are more interesting questions than the time to answer them. So I think that there are like a lot of open questions there. We were focusing on just some of them. So in general, like at some point, we decided that we will write it as a short review because we found that there is not so much literature. So we want to go into a like narrowing it down to the plasticity papers. Luckily, at the same time, the group for Kuipers, I don't remember her name. So they published that. I suggested you to read this paper.
DeVos, yeah.
Wrote like a very similar review, but it's much more broader. So likely you have two papers to read if you want to have a narrow focus on the plasticity or more broad about some mechanisms of this plasticity. And some questions which rose, but we decided maybe not to touch them because our review is not covering them, are as follows. So one question which is interesting is, and it comes from some papers done, is the sex-dependent effects of the psychedelics. So in most neuroscience research, we preferably use the male rodents, male mice, male rats, because they are easier to handle. A lot of models are like established for mostly males. So there is a bias towards effects of these drugs on male animals, not females. But in some studies which covers both male and female groups, there are some differences in the effects. We see some effects on females, but not on males, or on the other side, like reversely. So like for example, there is one paper by David Olson's group made by Cameron in I think 2018, and they found a sex-dependent effect of chronic dosing of LSD. And also one of the most extensive papers from Alex Kwon's lab, I think that the first author is Shao, and it was published last year in Neuron. They also found different effects in males and females. And these papers are not discussing these effects enough, probably because there is not so much to rely on, because there are like just a few papers finding different effects on males and females. So that's one very interesting thing. So how the psychedelics differentially affects males and females. That's pretty important question if you want to use them in the clinic. So we have to think if the female patients and male patients should have a different administration procedures, or maybe these compounds would work on just a subgroup of our patients. So that's one thing. The other question is the question of dose. So like it's a general broad issue. So for a lot of these compounds, therapeutic dose is quite close to the toxic dose. So in many cases we have to look for these effects. So for example, that's one of the cons of using the ibogaine. So the therapeutic dose is, if I remember, about 40, and the toxic dose start at 50. So like just, I don't know, 25% more than you need for the therapeutic dose may cause heart problems like heart failure, or starts to be neurodegenerative rather than neuroplastic. So a matter of dose is very important, and all these questions like how to administer this drug, how to test these drugs in animal models which have a different metabolism, and the doses and the routes of administrations are differs from what we use in humans. When it comes to the administration and the dosage, we know that a lot of these compounds were traditionally used as a mixture of compounds, such as ayahuasca. It's not clean DMT, it's a mixture of different compounds, and the studies on ayahuasca extracts give some different results than the studies done on the DMT alone. So that's also a matter of dose and then interaction of these. We need much more research in the field of dosing. We need a much more research in a field of time effects, as we discussed. Like, we don't know too much about long-term effects. When it comes to the time and dosage, we need a research on the microdosing, because there is almost no research. So there are like just a few papers, but very briefly mentioning like how the multiple low doses of the psychedelics actually work on preclinical models. So most of what we know about the microdosing are from the like people's report, which we know from the some of the last papers on the like users of the microdosing, that they are not very reliable, because it's very hard to put the prepare the placebo. People know what they are using, so actually we don't know. There are not so many papers about the clear biological mechanism undergoing microdosing, but it's at the same time, we know it's a growing trend in the society. So a dosage and the timing, sex effects, and the next thing, which I think is very important, and we were like going around it through this seminar, is what are the functional implications of the observed plasticity. So we may have a plasticity, but what does it what it means, like right. What it actually changes, and is it therapeutic, and if so, in what kind of setting, and what do we need to like how we need to prepare the experiments, and how we need to prepare the routes of administration in the therapeutic settings to actually have functional effects of these plasticity changes. Yeah,
that's right. And I would also add to all this, it's also really important to corroborate all these preclinical findings in humans. And it's notoriously difficult to study neuroplasticity in humans without having access to the brain itself. All right, so we have approached the end of the session. If anyone has a quick question, you can say it now. Otherwise, I would like to thank you all for joining us tonight.
May I ask something really quickly? Is that okay?
Sure, absolutely.
I was just really curious about why specifically, just like in a couple sentences, why study astrocytes and glia specifically, their effects, how they change in psychedelics. Specifically in the preclinical studies, if that made sense.
So you're asking why?
Yeah, why study that? Yes, exactly. You had mentioned it, and I just want to know what your thinking was.
Yeah, I think in general it's very important, because like we are in neuroscience, we are still assuming that the neurons are doing their work and they are alone there in the brain. But we know that there are like 10 times more glia around them, and we know that the synapses should rather be described in the textbooks as a tripartite synapse than just the dipartite synapse. So we don't have only the presynaptic part, which is releasing the neurotransmitter, and postsynaptic part, which is receiving it and using the receptors. But we have glia covering the synapse, and it is very important because it affects actually how much neurotransmitter is on the synapse. So it affects the excitability of the synapse, because this space in the synapse is very tiny, and depends how tightly astrocyte is covering it. We have changes in the concentration of the neurotransmitter. So we don't need more or less neurotransmitter. We can regulate it via the astrocytes, for example. Also, astrocytes have neurotransmitter reuptake proteins. So a lot of the neurotransmitters can be taken out from the synapse, decreasing the concentration of the neurotransmitter by the astrocyte itself. And what is interesting, like one astrocyte can cover like around 1,000 of synapses itself. So it means that if you have a change in just one cell, it means that you can regulate, like down-regulate or up-regulate the threshold of activity of 1,000 of synapses. So it's a rather like a network regulating effect, which is not so well studied yet. And as I said, like when it comes to the psychedelics, I haven't read any paper about the glia and psychedelics. So maybe there is some, but it's like less than a dozen probably.
I also have not. Yeah, it's an entire career basically of research just waiting there.
No, thank you so much. I appreciate that. Thank you both.
All right, thanks Raven, and thanks everyone for joining us tonight. Unfortunately, we don't have time for another question, but I would like to invite you to our next session on the 22nd of June. And I wish you all a pleasant night, and it was nice to see you joining us tonight.
Okay. Yeah. One thing. I think I may write my email on the chat if you would like.
Absolutely.
You can write me an email if you need some more. Explanations. Yeah, but yeah, because like in general, I am interested in the psychedelic research. I'm trying to manage it in my own lab. It takes time, so now I have a break in the experimental part because but I want to continue this because as we were discussing it today, it's very promising. And I think like if you look at it this way, that we have a general crisis in the psychiatric drugs. We have drugs which are not working on the real problems, and we know that we have more patients coming on than actual methods of therapy. So we need to have some revolution in the drug field. And I think like looking wisely at the fields, maybe not all, but a particular type of the psychedelics in the particular setting are the answer for the psychiatric crisis in the field.
All right. Thanks a lot for that addition, Kacper. And I wish you a pleasant night as well.
Thank you. Thank you all.
Yeah, goodbye.